The objective of this experiment is to observe microorganisms using light microscope, to view types of microorganisms and their structure, to observe different microorganisms and learn how to describe their morphologies, and to learn a great deal about microbial cells, including their structures and morphologies. One of the most important instruments used by the microbiologists is the microscope. The microbiologists can discover a great deal about the morphologies and structure of microbial cells with the use of the device. The combination of the ocular and objective lens produces the magnified image that we see. The ocular lens on the majority of microscopes magnifies objects by a factor of 10. The three objective lenses that the light microscope often have a low power, 10 times, high dry, 40 times, and oil immersion, 10 times. The product of the ocular, 10 times, and the particular objective lens that was used yield the instrument's final magnification. For instance, object can be magnified 400 times by using the ocular and high dry lens together. Learning how to use this instrument properly is critical to your success in the microbiology laboratory. Properly used, the microscope will open up the beautiful microbial world. We introduced for light microscope. It composed ocular lens, body tube, arm, objective lens, stage, illuminator, coarse focusing knob, fine focusing knob, and base. Adjusting the microscope. We raised the condenser to its highest position and adjusted the field diaphragm. First, we began with the low power, 10 times, objective lens for scanning larger specimens. Then, we adjust the distance between the lens and the slide using the coarse adjustment. And then, we focus using the fine focus knob while looking through the ocular lens. Lastly, we fine-tune the condenser iris diaphragm for contrast and resolution. Microscope is an instrument that produces a large image of small objects and allows us to observe an exceedingly close view of minute structure at a scale convenient for examination and analysis. Thus, with the help of light microscope, we were able to observe various types of organisms. The observations that we obtained were shown in the next slide. Bacterial flagella, observed with magnification 100. The structure was thick, rigid cell, and spiral shape. It has amphitheris flagella type and flagella present for locomotion. This bacterial was a gram-negative bacteria. Bacillus cereus, observed with magnification 100. It has rod shape and a toxic produced facultatively anaerobic gram-positive bacterium. Bacteria type gram stain observed with magnification 100. It was negative gram bacteria because of the pink color. The cell walls with thin layers of peptidoglycan, which is 15 to 20% of diameter. Hansenula was observed with magnification 100. It was positive gram bacteria with spherical shape. Bacterial capsule was observed with magnification 100 and the structure present is coccus shape. It has polysaccharide layer that lies outside of the cell envelope. Penicillium was observed with 10,000 of magnification and using this objective lens needed to add the oil immersion. The structure was a brush-like shape known as a penicillus that contained both branches and metallic. Mixed coccus gram stain were observed at 100 of magnification. The structure was mixed with coccus shape and positive gram bacteria.
Treponema was observed with 100 of magnification. The structure present in helical, coiled, corkscrew-shaped and has 6 to 15 micrometer long and 0.1 to 0.2 micrometer wide. This bacteria was negative gram bacteria.
The objective of this experiment 1B is to show the existence and the variation of microorganisms in the environment. Microorganisms can be found all over the environment. In a laboratory, for example, microbes are present on the air, clothing, tabletop, and equipment. These microbes often have an ability to infect pristine cultures. A culture used in a microbiology research needs to be pure. We saw the existence of microbes in the environment. Before we start, materials will be introduced. Sterile distilled water, nutrient agar plate, potato distress agar, test tube containing sterile swab. For table surface, the swab will be moistened with the distilled water through a septic technique. The swab will be brushed on the Na agar surface. Result for the table surface. After 24 hours, the Na surface of the table surface sample incubated, visible white spot appears. For air, first, both closure of the Petri plate will be open. Na Petri plate will be weighted for 30 minutes and PDA Petri plates will be weighted for 15 minutes. Both of the Petri plates will be inserted into the incubator for 24 into 48 hours. Result after 24 hours. White spot appear on the Petri plate that contain Na agar. In the PDA Petri plate, just a few white spots appear. For skin surface that using fingers, Petri plate will be labeled with a group name and the date of the experiment begin. Petri plate will be divided into two segments. The clean side and the dirty side. The dirty thumbs stand inside Na agar and the clean thumbs stand inside Na agar. After that, the Petri plate will be sealed and inserted to incubate at 37 degrees Celsius. White patches appear more on the dirty side than the clean side. Since it is a small organism that can be seen by a naked eyes and will often overlooked that it existed everywhere. The existence of the microorganism in a laboratory can be contaminated the pure culture and hence is the result of the experiment. Therefore, the objective of this experiment was to see the existence and the variation of microorganisms from few sources of the environment. In this experiment, aseptic technique can use to isolate the environment. Aseptic technique was a technique that microbiologists use for a variety of procedures such as transferring cultures, inoculating media, isolation of pure cultures, and for performing microbiological tests. The first part of the table surface, the results shown that there's a colony of microorganisms, or oftenly called as a germ, were present. The possible germs that can be found on the surface of table were E. coli, Salmonella, and more. Next part, which is for the air, we could show that on the neutron agar plates that have been left exposed for 30 minutes have microorganisms present. The reason was because microorganisms that exist in the air already occupied the Na plates were left exposed and have grown faster due to the sufficient nutrient and temperature. Meanwhile, for the potato distros agar plates, there were no microorganisms present. Supposedly, fungi microbes can be found in the PDA plates such as Aspergillus, Penicillium, or Rhinoceros, since these fungi microbes were commonly found in the air. No microorganism found on the PDA plates may due to the reason that they were not existed abundantly in the laboratory area due to not suitable for growth, and they grew slowly for 24 hours were not enough time for them to grow, and hence nothing present in the PDA plates. After that, for the skin surface, we obtained the microorganisms were present almost equally in the both clean and dirty segment. The result that we supposed to get were more microorganisms present in dirty segment compared to clean segment. It is impossible to get no microorganism present in a clean segment due to soap not being able to remove all the microbes from our hand. However, since we obtained the result in both segments, microorganisms present almost equally because of some error like not washing hand properly or unconsciously touching other area with the finger that later praise the clean segment and thus affect the result.